Hi, and welcome to Durham College's Student Academic Learning Services video on membrane transport mechanisms. By the end of this video, learners will be able to explain the function of cell membranes, differentiate between passive and active transport, contrast diffusion, osmosis, and facilitated transport, and contrast pumps and bulk transport. Looking at a cell, we can note that the cell membrane, sometimes referred to as the plasma membrane, is the outer wall of the cell. Cutting through that, we can see that there are a number of chemicals that make up the cell membrane. There are phospholipids that make up the phospholipid bilayer, proteins, carbohydrates, steroids, and various other chemicals. Again, the role, though, of the cell membrane is to separate the inside environment of the cell from the outside environment. Looking at the cell membrane from a slightly different angle, we can again see that the inside of the cell and the outside of the cell are separated by the cell membrane. The cell membrane, again, is made up of phospholipid molecules making up the lipid bilayer, and embedded within that lipid bilayer are proteins. Inside the cell, separate from the cell membrane and outside of the cell as well, there are a variety of chemicals. These include charged particles, water molecules, non-charged particles, and macromolecules. Non-charged particles can be found both inside and outside of the cell. At times, these particles may move through the phospholipid bilayer in order to enter or exit the cell. Water molecules are also found inside and outside of the cell. Instead of passing through the lipid bilayer, water molecules will move along special proteins in order to move outside or inside of the cell. Charged particles cannot move across the lipid portion of the lipid bilayer. However, as we'll see later, there are ways for them to get across the cell membrane. Macromolecules, being quite large, will also not enter through the lipid bilayer. It is important to remember that the system is very complex. There are many particles, many chemicals, both inside the cell and outside of the cell, as shown in this image here. In the end, for membrane transport, there are two major types of transport: passive and active transport. Passive transport are when chemicals move across the membrane from high to low concentration. No energy is required for this process to occur. On the other hand, active transport is when chemicals move from low to high concentration. And in this case, energy is required by the cell in order to move those chemicals. So in the end, membrane transport is broken down into one of two major categories, as we just saw: passive transport and active transport. However, there are also subcategories to each of these types of transport. For passive transport, there are simple diffusion, osmosis, and facilitated transport. For active transport, there are pumps and bulk transport. So let's take a closer look at diffusion. Diffusion is when non-charged particles move across the cell membrane from areas of high concentration to low concentration. Osmosis is when water moves from areas of high concentration to low concentration, but in this case they don't travel through the lipid bilayer, but through special protein pores called aquaporin. Facilitated transport is facilitated by proteins embedded in the lipid bilayer that allow for large macromolecules, like glucose in this example, to move from one side of the cell to the other. The protein will attach to the glucose, change shape, and allow it to be released to the other side. We can now compare diffusion, osmosis, and facilitated transport based on what's being transported, how it's being transported, concentration gradients, and energy requirements. For example, in simple diffusion, small uncharged particles like oxygen and carbon dioxide are being transported. In osmosis, water is being transported, and in facilitated transport, large particles, for example glucose, are being transported. In simple diffusion, the particles are moving through the membrane or through protein pores. Osmosis, the water is moving through aquaporins, which are protein pores specific for moving water. In facilitated transport, carrier proteins are moving the molecules. In each of these types of transport, the concentration gradient is always from high to low. Something else that these each have in common is that the energy requirement is that no energy is required. Let's look at the transport review again. Recall we have passive transport, which we just looked at, simple diffusion, osmosis, and facilitated transport. And we have active transport, which includes pumps and bulk transport. Now let's look at pumps. Specifically, we'll look at charged particles being pumped into and out of the cell. Proteins found in the cell membrane, for example, the sodium-potassium pump in this case, will bind to potassium and sodium and pump sodium out of the cell and potassium into the cell. However, this requires energy in the form of ATP. Bulk transport, on the other hand, is when vesicles are formed by invagination of the cell membrane, picking up a variety of chemicals, solutes, from outside of the cell and then bringing them into the cell. This process also requires energy. So in the end, we can compare pumps and bulk transport as well. What's being transported? In pumps, small charged particles. In bulk transport, bulk or large amounts of mixed chemicals. In pumps, it's proteins within the cell membrane. In bulk transport, it's in vesicles. The concentration gradient in this case is the same, from low to high and low to high in both cases. The energy requirement is also the same. Energy is required for these two active transport mechanisms. So in the end, we have this overview of membrane transport. We have passive transport, which is made up of diffusion, osmosis, and facilitated transport, and active transport, which is made up of pumps and bulk transport. Now that you've watched this video, you should be able to explain the function of cell membranes, differentiate between passive and active transport, contrast diffusion, osmosis, and facilitated transport, and contrast pumps and bulk transport. Are you still confused? Do you have any more questions? Please feel free to contact us to make an appointment with a SALS science specialist. You can reach us by phone at 905-721-2000, extension 2491, or by email at sals@durhamcollege.ca. Please help us gather feedback to improve the service we provide in student academic learning services. If you're viewing this presentation online, please email us any suggestions you have to improve the experience at sals@durhamcollege.ca.